All right, let me just make sure that I've got this on mute and then we'll get underway here. Okay, awesome. Thanks everyone for your patience for last week. I ended up coming over just to make sure that no one showed up to class. Apparently everyone got the message because no one came to the classroom except me. But I had to start getting ready for my procedure at noon, and I'm like, I don't want to be in the middle of class trying to figure things out while I'm starving and whatever. So anyway, how many of you had a chance to watch the relevant videos for last Thursday? Awesome. I saw a few of you submitted either titles or one person submitted an actual full project for review. And so far, so good. If you're just submitting a title and a quick summary, I think I'm giving you 20 points out of 100. And then on the person who submitted a mostly done project, I gave a grade that, you know, can still be upped, but looked pretty good. I was pretty happy with what I was seeing there. Continue to work on that. Make sure that I'm not leaving steps off, but I believe next week is the first exam, and I have it set for the 3rd, which is that Thursday, but it'll likely open on the 2nd or 3rd, and then it'll close on probably Saturday night at midnight. I'll try as much as possible to always have the quizzes close on the weekend. That's a double-edged sword. It's nice because it gives you a chance, you know, if you've got a crazy week, you can still get everything done, you know, on the weekend. But the risk is if you run into any technical snags, I'm not as good about checking my emails on the weekends as I am during the week. So you may have to give me a call on my cell phone if it's something that's of immediate urgency. I have on here project 1 checkpoint and details. All I'm looking for is a title. If you don't submit a title, that's not the end of the world. But if you want any feedback on it, that's your opportunity to get some feedback on your project. But the project 1's going to be due about the 2nd week of March, right before spring break. All right. Any questions related to project 1 before we jump into our meristems and roots and shoots? Yes.
It can be pretty much anything. Like I don't really understand like what kind of topic.
Okay, so the question was, can it be pretty much anything, or you know, do we need to make it a little bit more defined than that? A little more defined than that, but not much. As long as it relates to plant growth, development, or physiology, I'm pretty open to it. It could even be something that's that affects plant growth and physiology. Say you want to do sunlight or something like that. That's not, you know, strictly a plant process, but is something that affects plants. You know, if I can see a connection to plant science, usually I'm going to be pretty comfortable with it. You know, if you're planning to major, if you're majoring, for instance, in genetics, if you want to do it on plant breeding or plant genetics, I'll be pretty flexible on that. But, you know, I need to be able to see some connection with the plant somewhere in there. But beyond that, you know, I'm not going to assign topics. Other questions?
Can it be about like misbench or something? How do they?
Okay, question, could it be something like a mist bench where it's a management option with, you know, with the plant system? Sure, just make sure that it ties back to, you know, mist benches and, you know, ways to use them with plants as opposed to mist benches. And, you know, I set it up for a sauna so that I could hang out during the winter calendar type thing. So yeah, as long as I can see the connection, I'm going to be pretty flexible on that end. Again, this is a communication literacy assignment. What I'm looking for is you having an opportunity to become a better communicator. But I wanted to have some connection with plants because it is a crops class. How many of you were pretty happy with how my one pager turned out with only about an hour of work on it? Okay, a couple of you not. I'm like, come on, I was pretty proud of it. I may even have I think I uploaded a PDF of it here. I did project. Actually, I don't see it either. I think this is the old one. Yeah, this is the old one. I'm not as happy with the old one as I was when I originally made it. Maybe I'm getting better myself. I don't know. But I'll try to upload the version that I did for the actual video for all of you as well. So we'll sort that out. All right, with that said, today's lecture, and again, I try not as much as possible just to rush through lectures. With that said, I was looking at the syllabus, and this lecture is one of those that we can cover, I think, fairly rapidly. And I want to make sure that we get all the way through it today. It's 30 slides, so you would think that you could get through 30 slides without feeling like you need to be rushing things. But when you get to be old like me, it turns out that you can talk a lot about individual slides. So I'll try to move things along fairly quickly just so that I can make sure that we get into plant growth and development on Thursday. And we have time to actually work on doing growing degree days, because that's something that I want everyone to have a chance to feel comfortable with. The reason I added this, I actually added this about 5 or 6 years ago, because I discovered most of you don't actually, if you're not a horticulture major, you're not taking classes that in plant anatomy or plant structure and function. How many of you who are not horticulture majors have taken a class in plant anatomy? Okay, a couple of you have, but I know a few of you haven't. And so I thought it's useful to talk about the anatomy for one day, and then we can move in and we're all speaking a common language. But I'm not obviously going to go into as much detail as if this was a plant anatomy class. We're going to spend a fair amount of time talking about meristems, because in my mind, when we're talking about plant growth, we're talking about meristematic growth. We'll talk about some of our primary meristems, which include your apical meristems, which tend to be at the tips of the plant. We'll talk about axillary buds just a little bit. We'll talk about intercalary meristems. And then we'll talk about secondary meristems, which are usually lateral growth within a plant. And we'll define all of that as we go. We'll talk about general plant tissues, and then we'll talk about root stems and leaves, and then we'll be done. For whatever reason, every time I get to this lecture, I'm reminded of an old science or an old writing book that was called Eats, Shoots, and Leaves. And it's about the old panda joke about the panda walks into the bar, shoots a guy and walks out. And the bartender's like, "What are you doing?" And the panda says, "Well, it says right in here in the book, eats, shoots, and leaves." And so, you're going to be forced to listen to dad jokes in this class because I'm old. So meristems, if we needed to define them, are going to be regions of undifferentiated tissue that are responsible for growth in a plant. When I say undifferentiated or dedifferentiated, what we mean is tissue that can essentially be turned into just about anything. And so meristematic tissue is really cool because it can grow and it can expand and it can develop. And then it can actually turn into specialized tissue that has a specific function within the plant. So meristems are going to be regions of plant growth and cell division within the plant. This slide here was actually done here at Texas Tech by an undergraduate student, or I think actually a master's student by the name of Craig Bednars, who many years ago was a student here at Texas Tech. And then later was my advisor at the University of Georgia. And he did this really cool slice and slide while he was here. And all I did was colorized it a little bit, added some labels, and slapped it into a publication that we did together called Cotton Growth and Development. But you can see primordial tissue here. And even early on in this process of development, you can start to see the development of some of these specialized tissues within the plant. So meristems are going to make new cells for primary and secondary growth. The reason I put a 1 degree and a 2 degree is in chemistry, 1 degree means primary, 2 degree means secondary. I'm not going to ask you on the test, what does a 1 degree stand for? But I was too lazy when I put together this slide to type out primary and secondary. Now, after many years, I've spent more time explaining it than I would have just typing in primary and secondary. But meristems are going to make cells for these 2 types of growth. Now, most types of meristems that we're going to talk about are actually focused on primary growth. Sarah has the disadvantage of having asked me a math question, so I remember her name. So today, as I go around, if I goof up on your name, or if I ask you for your name, please tell me. Every time I'm going to get a little bit better, and eventually I'm going to more or less know everyone's name. But if my children are any indication, that doesn't mean I'm going to call you by the correct name in the process, because I don't even call my own kids the right name. Extra bad when you have pets, because you start calling the pets kids' names and the kids' pets' names. We won't get into that. So, Sarah, how would you describe plant growth? How do you know that a plant has grown? Let's say that you look at your plant in a pot one day, you come back a few days later and you say, oh yeah, that plant's grown. What difference are you seeing in that plant?
Usually it's like taller.
Okay, great answers, both. You can pass it now. She said, usually the plant's taller or the roots have expanded. And those are both very good examples of primary growth. Anytime a plant gets taller, anytime the roots grow longer, anytime the roots branch out, all of those are going to be examples of primary growth. And most of the growth that we can visually see in a plant, at least over the short term, is going to be primary growth. And so apical meristems, well, let's see. And is it Carrie?
Yes.
Okay, Carrie. You didn't know that this was going to be an English exam today. What is an apex? If you're climbing to the apex of a mountain, which part of the mountain is that going to be?
It's like the tip.
Okay, it's the tip. So an apex is the tip, and therefore apical meristems are going to be at the tips of the plant, not only at the top of the plant, but also at the tips of the roots. And so apical is simply, they took the word apex, they turned it into a relative term, and they're saying these are the meristems that are at the apex or the apices of the plant. They're often enclosed by leaves or protected by a root cap. And you can see, here's our apical meristem right here in the middle. And you can see we already have leaves or leaf primordia that have formed around this apex in a bundle to protect it, because if you damage your apical meristem, your plant's going to have all kinds of problems. Yes?
When does the meristem differentiate? Like the cells?
Oh, great question. When do the cells of the meristem differentiate? Generally speaking, and going back to this slide, as you get into the intercalary meristems, which is kind of in between the apex and the mature plant, the cells at that point are already differentiating into more mature tissue. So if I were to give a general back of the envelope rule, I would say that your apical meristems are more or less undifferentiated. Your intercalary meristems have already begun to become differentiated so that by the time you get down to the actual plant below the apical meristem, it's already differentiated and it's more or less locked into place. Great question. And it's Alexa? Okay. All right, awesome. You can pass it, Terry. Great job. So apical meristems, and you also have, if you look in the junctions of plants, places where the plant's going to form branches, places where it's going to form flowers, you're going to see things called axillary buds. These axillary buds are also primary growth mechanisms where you're going to be producing something that expands out and gets taller or longer in the process. So apical meristems and axillary buds are both going to be very similar in nature, and they're both going to be types of primary growth. And then, as we just talked about, intercalary meristems are the in-between meristematic tissue between completely immature tissue and completely mature tissue. And these are also considered primary growth, but they're also considered growth that's kind of in between in the plant here. Now, interestingly enough, and let's see, Blake, how far from the tip of this plant is this intercalary meristem?
Possibly.
Okay, so that great answer, centimeters to inches. So interestingly enough, and this goes back to the question that Alexa asked, if the primary growth is occurring centimeters to inches behind the top of the plant, and typically, and again, you'll hear a lot of cotton examples in this class because, you know, I know a lot about cotton because I look at it about 5 hours a day during the summer. But this is applicable to a lot of different kinds of crops. So when you hear me say cotton, unless I specifically say, and this is something that only occurs in cotton, assume that it's something that occurs in other plants as well. A cotton plant, we actually spray plant growth regulators on it to keep it from getting too tall. But those plant growth regulators only really act on the parts of the plant within about the upper 3 to 4 leaves within that plant. So, you know, the upper few centimeters or inches of that plant, you know, it's not going to have any effect on the rest of the plant that's already mature tissue. And so the same thing occurs with trees. You know, when a tree grows, it's not all growing. It's really only the tips of the trees that are growing upward and outward at any given time. Good job, Blake. Go ahead and Alexa, you can either answer a question or pass since you asked a good question. All right, brave person. Okay, so we talked about how meristems go upward and outward, you know, upward and, you know, expanding, something that's relatively easy to measure. What about a tree? I said that a tree is not going to expand and get taller like in the middle of the tree. It's all happening at the tips. And yet, if you go out and look at a tree today and then come back in 20 or 30 years, that tree has gotten wider over time. So what's going on with that?
It's growing layers of secondary growth or bark.
All right, good. It's growing layers of secondary growth or bark. Great answer. Essentially, what that tree is doing is it's forming every year, it's forming a layer of xylem and phloem. at the outside of that tree trunk, and it's using that as vascular tissue. And then the next year, the xylem, stops growing and essentially it starts over. And so the rings of a tree are actually that process of growing outward on a yearly basis. And so this is, as Alexa mentioned, what we call lateral meristems. And lateral meristems or secondary growth are just the outward growth, particularly of perennial plants, but you also see some lateral growth in annual plants as well. But it's just that outward expansion. So apical meristems, axillary buds, and tertiary meristems are plants growing taller, plants putting roots down. Lateral meristems are anything that's going to be secondary growth, which is the plant getting wider as opposed to taller. Any questions on that? Okay, good. The way I remember that is, when I was a teenager, I saw a lot of primary growth because I was getting taller rapidly. And now that I'm getting older, I'm seeing a lot of personal secondary growth, unfortunately. There are 3 types of general plant tissues that I want all of you to be aware of. Number 1 is just called ground tissue. This is just general all-purpose plant tissue. Ground tissue is anything that doesn't have a specific role and isn't differentiated into something unusual or unique. So that brings up the question, let me see, is it Lee? Okay, Lee. Here I'm looking at the upper surface of a leaf here, and right here I see a leaf hair sticking up right here. You think a leaf hair would be a type of ground tissue? Why or why not?
I'd say, what was the question again? Is it?
Yeah, would you consider a leaf hair ground tissue based off of the definition that ground tissue is just typical plant tissue that hasn't been specialized for a certain task?
No, I'd say, I think it's specialized.
Okay.
So it's not.
Lee says that it's specialized. And he's correct. So in this case, ground tissue might be individual tissue or cells within the plant, but as soon as you've specialized it toward a specific strategic goal within the plant, it's not going to be ground tissue in them anymore. In this case, and you can pass it now to Spencer if you'd like. Great answer. In this case, leaf hairs would be a specialized type of epidermal tissue. Spencer, how about this tissue that's floating around here in the apical meristem? Would this be considered ground tissue?
No. You talking about like the xylem, phloem?
Oh, great question, or great answer. So I'll count that as a good answer. You can pass it. If it has differentiated into xylem and phloem already, it's not ground tissue anymore, it's vascular tissue. However, if it has not yet differentiated into one of these specialized types of tissue, it would be considered, in this case, ground tissue. So sometimes it will be ground tissue, sometimes it won't. But generally speaking, if you look in the apical meristems, generally most of that tissue is going to be ground tissue because it hasn't strictly defined itself as a specific type of tissue. Yes, Alexa.
Why are those ones on the edges called fruiting branches?
Great question. The question was, why are these ones on the edges called fruiting branches? Well, again, going back to cotton, because cotton's what I know. Cotton has 2 types of branches that it produces. One are called vegetative branches, and these are branches that are formed generally late in the season, and they look just like a cotton plant that's kind of grown off the side of the existing cotton plant. Whereas fruiting branches form this specific type of structure where it grows out, it puts up a flower, it changes direction, it puts up another one, etc. So in this case, all we mean in this case, and this one is specific to cotton, just because when I was putting the slide together, I was pointing that out. These are specifically branches that are going to grow just like general fruiting structures on the plant, as opposed to just making a big long branch with lots of other branches coming off of it. Boy, good questions, everyone. I'm almost going to forget who has the microphone, but I didn't. It's Taylor. So we've got to have a question for Taylor. Let's see. Taylor, we've talked about ground tissue. We've briefly touched on epidermal tissue. Vascular tissues are transportation tissues or plumbing tissues within the plant. Can you give me a couple of examples of types of vascular tissues that we've brought up in class today? All right, good. The xylem and the phloem are the two major types of vascular tissues that we tend to think of. Good. All right. Let's see. Is it Avery? Okay. Awesome. So these are the three general types of tissues that we see in plants. Now, tissues can be specialized above and beyond these three general examples here. So, for instance, if we're looking at this epidermal tissue here, the epidermal cells that make up the surface of a leaf are going to look substantially different than the epidermal cells that make up the leaf hairs, or the epidermal cells that make up the stomata. So you can further differentiate, but I mainly want you to be aware that there are these three general types of tissues that you see often in plants. And, this allows a plant to make itself into something that can be efficient. So now let's kind of chop the plant up into roots, stems, and leaves, and we'll talk about each of those individually here. Roots are going to have a few different purposes. They hold the plant in place. They're going to absorb water and nutrients. And some types of roots can also store carbohydrates and other types of energy compounds for the plant. Avery, that actually brings up a good question. Can you think of a couple of types of plants that actually store energy in their roots? Okay, carrots. Carrots are a great example of a plant that stores a lot of energy in its root. What does a carrot taste like? Yeah.
I don't know.
Wow. Do you not eat carrots because you don't like the taste? Okay.
I don't eat the carrot.
Okay, so Avery says the carrots taste bad. If you had to judge, would you say that carrots tend to taste sweet or sour or salty or spicy? Neutral flavor, yeah. And again, we kind of get ripped off here, only in that if you grow a carrot in the garden, generally speaking, that carrot's going to be pretty sweet. But if you buy a carrot in the grocery store, they tend to be a little bit more starchy because it's cheaper to produce carrots that aren't as sweet than it is to produce carrots that are sweet. So where am I going with that? Well, starches and also sugars are methods of easily storing energy in the root. So storage roots are going to tend to be large, like a carrot. They're also going to tend to often be either sweet or starchy. Good job, Avery. You can go ahead and pass it now. Let's see. Is it Megan? Caitlin. Sorry, Caitlin. All right. So, Caitlin, we've knocked out carrots. I have sugar beets up there. Can you think of another type of, I guess, storage root that you might see at the grocery store? Okay, any type of root vegetable, such as a sweet potato. Good job. By the way, interestingly enough, Irish potatoes or regular potatoes are actually a type of storage stem, which is kind of an unusual, I guess, plant and anatomical feature. But it's interesting that you bring up potatoes. I have one of my former graduate students who is a potato agronomist, and he is also a sweet potato agronomist. And I asked him, so, potatoes, you're growing it for a storage stem or a tuber. Sweet potatoes, you're growing it for a storage root or a tuberous root. I said, what's the difference in trying to grow sweet potatoes versus potatoes? And you know what he told me? He said, you grow them pretty much the same way. So interestingly enough, whether it is a potato or a sweet potato, you're still going to work toward the same goal, which is putting as many resources into either the root or the tuber as possible. Great job, Caitlin. You can go ahead and hand it back now. So are you Megan? Morgan, okay. Well, I'm just going to keep guessing until someone's Megan. So, all right. Let's see. So, as Caitlin mentioned, anything that tends to be a root vegetable is going to be fair game in terms of having a storage root. We tend to see two different types of rooting systems, generally speaking. We have fibrous root systems, which tend to have masses of similar equally sized roots, and we have taproot plants, which tend to have one main root and a lot of smaller roots that branch off from it. So, Morgan, would you say that you tend to see fibrous roots more often in grasses or more often in broadleaf plants? Okay, that is correct. So generally speaking, if you see a plant with a fibrous root system, it's usually going to be a grass. And I guess the second question would be, before you hand it off to Morgan, you tend to see, if you have a broadleaf plant, does it tend to have a taproot or does it tend to have a fibrous root? Good. So generally speaking, your dicots or your broadleaf plants are going to have taproots and your grasses are going to have fibrous roots. I hesitate to say that that is a 100% rule, only because a lot of your dicots, even though they have a general taproot system, if you pull them up, they're actually going to not look significantly different than a plant with a fibrous root system, even though you can generally track that taproot down in the plant. Good job. Okay, go ahead and pass it along. Let's see. Megan, let's see, is it Grayson? Okay, sorry, that's right. Hey, Nava, remind me your name. Seth. I'm sorry, Seth. For whatever reason, and I know that I should know your name, but it bounces back and forth in my mind. All right, Seth. Actually, you may not have a question on this slide, so you can relax. If you slice a root from the very tip up to where you get into the mature tissue, you're going to tend to see three different regions before you get to mature tissue. The root tip is going to start out with a root cap. And the main purpose of the root cap is just to protect the meristematic tissue. And what happens is you produce root cap and it tends to get sloughed off over time because it's in contact with the soil, and the soil is pretty rough on the roots. And so the plant actually has to expend quite a bit of energy just in continuing to produce root cap tissue that can be sloughed off. Behind the root tip is going to be a region of elongation. And elongation is going to occur due to two things. Mitosis, which is the splitting of cells, and cell elongation, which is due to pumping water into the cells. And okay, here's a question. Thought you weren't going to have a question on this slide, but you are, Seth. what type of a balloon makes the best balloon for making balloon animals? No, just shape wise. If you're making a balloon animal, are you going to use a round balloon or are you going to use a tube shaped balloon? All right. Plants use the same general strategy in terms of their cells, which is the cells tend to be, they start out kind of square shaped or a little bit rectangle shaped, but then they expand like a tube and they become longer in the process. So if you want to imagine what cell expansion looks like, imagine taking Seth's balloon animal balloon, filling it up with water, watching it expand lengthwise. That's essentially the same process, except that if you do it with plant cells, they're little bitty and you can't then, try to attack your children with long elongated cells like you could with a long elongated water balloon. Not saying that I've ever done that, but theoretically you could. All right, good job, Seth. Okay, so Grayson, the real Grayson. All right, so that's the region of elongation. And then you've got a region of differentiation. Remind me again, Grayson, anytime we said the meristematic cells are undifferentiated. So what's happening to these cells when they're differentiating? All right, good. They're choosing what type of tissue they're going to be, and they're locking themselves into being a specific type, whether that is epidermal tissue or vascular tissue or other just general ground tissue. They're essentially assigning themselves toward what other function they need to be based on where they're located within the plant there. And so you see the development of specific tissues. You see the epidermis. You're not going to need to know cortex or endodermis or still or cambium. Just be aware that as you get into this more mature tissue, it's now differentiated into specific types that allow the plant to be efficient in what it's doing. Good job. You can hand it to Carson now. Just Carson, right? Okay. So, interestingly enough, okay, I apparently, I thought this slide was going to be next, but I have this slide next. If you actually look at plant roots, especially imagine a big taproot plant, how much water is that plant going to absorb through its taproot? Yeah, you'd think that maybe that taproot's really going to town because it's so big, right? The reality is the only part of that taproot that's actually absorbing anything is the very tip of it. All of the work's being done by the root tips out in the other parts of the plant. So here we have this fibrous root, and I have these circles around the very tips of the root. That's where all the actual absorption is taking place. Whether it's a taproot or whether it's a fibrous root, all of the work is actually being done right within a few centimeters of the root tips. If you look closely at the root tips, you're going to see a bunch of these hair-like protrusions here, which are actual individual cells that have the sole purpose of taking up water, taking up nutrients. Good job. You can go ahead and hand it back now. Is it Hadley? George. Okay. Yeah, I don't see the Finken binders here today. Sorry, George. The Finken binders tend to sit toward the back, and they tend to be bigger guys, and so I thought maybe you were the younger of the two. So George, sorry about that. All right. So look, how's your eyesight? Good. Okay. So George, this is a cross section of a root here, and I circled a root hair here. How many different cells does it look like this root hair is actually broken up into? That's kind of a difficult thing to see from the very back, so I apologize. Yeah, it's actually, like George said, it's only one cell. And so these root hairs are actual individual elongated cells that have expanded off of the plant to try to capture water and nutrients out of the plant. Good job. You can go ahead and, yes, Alexa.
What do they use hairs for?
What do roots use hairs for? The smaller the volume of an individual cell, the easier it is for it to uptake larger quantities of water and nutrients, and also the easier for it to navigate through the path of the soil particles that are in the soil. And so root hairs are able to go places that the rest of the root is unable to go, and they're able to absorb water and nutrients that the root would actually not be able to absorb. So great question. That actually brings up an interesting question, not to get too far off track here, but have any of you ever heard of root hyphae? So root hyphae are actual fungi that have formed a symbiotic relationship with plants. And these root hyphae will attach themselves to plant roots, and they'll siphon off carbohydrates from the plant roots. But the plants are okay with that because the root hyphae are even narrower and longer than the root hairs. And these root hyphae will actually extend out into the soil and capture water and nutrients that the roots otherwise would not be able to capture. So it's advantageous to the plant, and it's also advantageous to the fungus in this case. So short answer is root hairs go where the rest of the roots are unable to go. And the long answer is you can actually have mechanisms by which plants can be even more efficient at grabbing water and nutrients than you could with root hairs. Great question. All right, let's see. Remind me your name. What's that? Rogelio. Okay, thanks. Do you have the microphone now? Okay, awesome. So, interestingly enough, up here with this cross section, you see a few root hairs. Down here, we have a colorized scanning electron microscope of a young barley root. This entire bundle are root hairs. So you can actually get a few, or you can get up to 2,500 root hairs within a centimeter squared of the root surface here. So what do you think affects how many root hairs you're going to find on an individual root, Rogelio? Okay, amount of nutrients in the soil. Good answer. Go ahead and pass it on. Is it Calvin? Okay, Calvin, any other ideas? So quantity of nutrients, what else might affect the number of root hairs? Okay, water. Water also affects the number of root hairs. Good job. We're going to keep going until we run out of ideas. You know, this is like Family Feud. We're knocking out answers, and so pretty soon we're going to get to that one strange esoteric answer that no one knows. Let's see. I should remember your name, but remind me your name. Will. Oh, sorry, Will. Okay, Will. So nutrients and water affect the quantity of root hairs we have. What might also affect your number of root hairs? Okay, the growing conditions such as temperature and sunlight, those can also affect root hairs. So good job. All right. Let's see. I know your name is close to Annika, but it's not Annika. Enika. Okay. Enika. So we've now eliminated nutrients, water, and other general growing conditions. Can you think of anything else that might affect the number of root hairs on a root?
The plant itself.
Okay, the genetics of the plant itself. So some species are going to have different numbers of root hairs, generally speaking, than other species. Wow. So those are all good answers. Let's see, and it's mahogany, right? So should we keep trying to come up with additional, or does it feel like 4? Okay, mahogany. Okay, good. The general health of the plant and the root generally. Good job. All right. Okay, soil texture. Great. All right. And I'll be honest, May, if you want to keep going, you can. We've pretty well knocked out all the ones that I know. Okay. Okay, if it needs more stability, yeah, I think that is a reasonable answer. So, wow, great job. Now we've come up with an answer that I hadn't even thought of. All right. Is it Brandy? Abby. Okay, sorry. So who's Brandy? Okay, sorry, Brandy. I should have known. I should have known that already. Okay, so Abby.
Would it be how old the root is?
Okay. All right, good. So older plants are going to have different numbers of root hairs than younger plants are. Generally speaking, and you'll notice that this SEM shows a young barley root with root hairs. Generally speaking, as plants start to get into flowering and fruiting, they tend to cut back on their roots, and as a result, they tend to cut back on their root hairs. All right, great job. Okay, Randy, I'm going to let you off the hook because I'm running out of ideas, and if I'm running out of ideas, eventually someone's going to be put in the untenable position of trying to answer a question that I don't know an answer to. And if I don't know an answer, it's probably not fair to make you come up with an answer. Generally speaking, roots are going to be very dynamic compared to the rest of the plant. You go out, well, when you go out from one day to the next, what name a type of plant that you might see on a daily basis?
Cotton.
Okay, cotton. So Brandy goes out on June 25th and she looks at her cotton plant. She comes out on June 26th and maybe that cotton plant has grown just a little bit, but by and large, does that cotton plant look very much different from one day to the next?
No.
Not really. May have grown a couple of centimeters. It may have started to put on a couple more leaves, but those leaves take a few days to develop, so you're not going to see major changes from one day to another. The roots, on the other hand, are going to be changing rapidly, and roots can actually change within a couple of days quite a bit. These are some side pictures that they took of corn roots over just about a 4 or 5 week period. In fact, this is 2 weeks, this is 7 weeks, so it was about a 5 week period. You see dramatic changes in the roots over that period of time. And yet, anytime you see plants growing, or we talk about plant growth, we almost never talk about roots, even though roots are the most dynamic growth that you tend to see in a plant. So do you want to answer that, or do we want to hand that off to Elizabeth? It is Elizabeth, right? Okay. If it wasn't Jeremy, you were going to be Elizabeth.
No,
So, okay. So Elizabeth, if roots are so dynamic and diverse and change so much, why don't we actually show much with them? Why don't we do a whole lot of research on roots compared to the rest of the parts of the plant?
I don't know if we can answer that.
That's kind of a difficult one, isn't it? So I'll throw it out here just from a practical standpoint. If I want to look at my cotton plant that Brandy's been checking out on a daily basis, and I want to see how much the roots changed from yesterday to today, what do I need to do in order to look at the roots? Yeah, I'm going to have to dig it up. How do you think that plant's going to feel when I try to put it back in the ground? Not great. Exactly. So generally speaking, the big limitations we run into with roots are, as Elizabeth said, that first of all, they're really difficult to measure in the first place. And number two, you tend to damage the plant when you're looking at them. So a lot of root work that's done is done through a few different types of methods. One is you look at it in hydroponics, where you can just lift up the container, see how the roots are doing, maybe even take pictures of them and drop it back in and the plant's happy. Sometimes they'll actually use like plexiglass sheets, put it down, and they'll keep it in the dark, and then they'll actually, uncover it, take a picture of the roots, cover it back up. Now that's not perfect because the roots aren't necessarily going to grow the same if they're running into a plexiglass sheet as if they're just growing in the soil. But we use all these methods, and we also tend to just kind of ignore them as much as possible. All right, great answer. Go ahead and hand it up to Jeremy. Well, Jeremy, the good news is we made it all the way around the class. The bad news is we still have a solid half hour, so you're probably going to end up answering like 50 questions. I don't know. Moving on to stems. Stems are going to vary a lot among plant types, but they're going to share general morphological characteristics, which are as follows, which this is pretty much what I want you to remember about stems. Here's what I want you to remember about stems. Stems have things that we call nodes, which are places that you form axillary buds. And they have internodes, which are, what would you say internodes are, Jeremy?
Between like the node and the next node. That's
Good. So the internode is simply, as Jeremy said, the space between the two nodes. So think of nodes as being places where you're going to be forming buds, and internodes as being places that are in between that allow the plant to elongate. Good job. Oh, no, you're not. No, I'm just kidding. You can pass it back over to Sarah. Maybe we'll see if we can get another round around. And, just be warned, Caitlin and Morgan and Seth, I'm just going to call you Megan because I'm locked into that name right now. So be prepared. So stems are going to form vascular connections from the roots up to the leaves, and they're going to consist of nodes and internodes. If you take a plant stem and if you slice it crosswise, it's going to look different depending on whether it is a monocot stem or a dicot stem. This one on the left is a dicot stem. You'll notice that it has these nice rings of tissue around the outside, whereas a monocot stem, like grasses, are going to tend to have their vascular tissue just kind of in random bundles throughout the tissue. So be aware of this. This also brings up a question. Sarah, how do you do a cross section of a plant stem without killing the plant? That is a good question. The long answer is you can hurry and put it back underwater, or put it in an aqueous environment, apply a little bit of oxen, and it'll reroot and it can grow. The short answer is that most of the time you just throw it in the trash and just kill it. So I've probably told you this. If I haven't told you this, I probably told my 1321 class does. People think that I become a crop physiologist because I like plants, but the reality is I became a crop physiologist because I like to kill plants. I'm a serial plant killer. So, you're allowed to do things to plants that you're not allowed to do with any other, or with many other types of organisms. So, if you were to see these on an exam, an easy way to think of it is that dicot stems are going to have a ring around the outside, which is going to be the vascular bundles. Monocot stems are just going to have them scattered around. All right, you can go ahead and pass this, Sarah. There are several types of specialized stems. Stolons are going to be above ground horizontal stems. Carrie, can you think offhand of any types of plants that tend to produce stolons? Okay, pumpkins are a great example of a plant that produces specialized stems as well, and they'll produce tendrils as well, which are specialized stems. Generally speaking, the way I define a stolon is it goes out and then it'll reroot down at other spots as it goes along. But it's not a bad answer. That's actually a pretty good answer. Yes.
Example being strawberries.
Okay, strawberries are a great example of a plant that I think of when I think of stolons, because they'll actually send out a stolon and they'll branch out and they'll put down another set of roots and off they'll go again. A lot of types of grasses have stolons. A lot of types of grasses also have rhizomes. Good job, Carrie. You can hand it back to Blake now if you'd like. So stolons are above ground horizontal stems. Rhizomes are below ground horizontal stems. They have relatively similar functions. Some types of plants will actually do both stolons and rhizomes, but the purpose is for them to be able to propagate vegetatively. So what does propagate mean, Blake?
Reproduce.
Okay, reproduce, or to, to expand their holdings, so to speak. Good job, you can handle it. So they use vegetative propagation or movement to expand themselves. Sorry, Blake, I should have asked you this before we passed it. Are those plants that are propagated vegetatively, would they be kind of like having the plant genetically having children, or would they be kind of like the plant just genetically still being itself?
I'm gonna ask a clarifying question.
Okay.
I haven't heard this, I guess, adverb used before. What do you mean by vegetatively?
Okay, vegetatively means anything that is the plant growing and turning into another plant without going through the seed process.
What was your question?
Would this vegetative expansion be like the plant having children, or would it be like the plant growing additional toes?
I would say additional.
Okay, yeah. So vegetative growth, such as with stolons and rhizomes, would be the equivalent of a plant just growing additional toes or additional plant parts and expanding. If you take a sample of these plants, they're going to be genetically identical to the mother plant, because essentially just a plant that's expanding itself over space and distance. We also have tillers, which originate from axillary buds. A lot of times you'll see tillers branch out from the base of the grass plant. You've got shoots and tendril, or twining shoots and tendrils, kind of like Carrie was talking about. Bulbs are interesting because here's a stolon, here's a rhizome. I should have been showing you these pictures. In fact, I'll just skip the rest of this slide and just jump into the picture. So strawberries are a great example of stolons. Here are some rhizomes for quackgrass, which are growing under the soil. Real quick, remind us, Alexa, how do we know that these are underground stems and that they're not actual roots?
Because they still have xylem, or no, well, that, I guess. that
Yeah,
makes
roots
sense.
have xylem and phloem as well,
Um,
but they do have something that sets them apart. It goes back to this slide.
nodes.
Okay, the big difference, the way that we tell the difference between a rhizome and a lateral root is simply that these rhizomes are going to have nodes and internodes on them. And so from a phenology standpoint, yes.
So what you call the offshooting roots, just like lateral?
Great question. So what do you call roots that offshoot laterally? Lateral roots. So lateral roots in many ways can look a lot like rhizomes. It's just that if you take them out and parse them and look at them closely, they're not going to have any nodes on them. So by the way, here's a node and here's a node and here's a node. So that's how you tell the difference. Good job. All right, go ahead and hand it back to, Lee, right? Okay. Let's see. Here's an example of tillers. They look like additional plants coming out from the same seed, but they're all connected to that same mother plant, and they're connected when you get down close to the seed. in the plant. A lot of types of grasses are gonna have tillers. Tendrils tend to be twining type stems. Lee, are tendrils going to have nodes and internodes?
No, they won't.
But how do we define a stem? Okay, so Lee is correct on both accounts. Tendrils tend to have one node at the bottom, and then they grow a really, really, really long internode, and it twines around, and you may not have another node on the end of it, but they are connected to nodes. So you're right on both counts. Good job. Go ahead and hand it to Spencer. So help us out here, Spencer. Would a bulb which grows underground be a root or a stem? All right. Are you sure?
Pretty sure.
Pretty sure. All right. It was actually a trick question. The reality is a bulb has both. The very bottom of the bulb, underneath it, like right in this area, you have little roots coming out of it. The rest of the bulb is actually a very compacted stem, and it actually the layers of it are actually modified leaves. So a bulb is actually going to have roots, stem, and leaves all packed into one combined thing. So you'd have been right and you'd have been wrong no matter how you answered. So
I just considered it a modified.
yeah, it is a modified root, but it also is a modified stem with modified leaves on it. Elizabeth.
So like on an onion, what would the shoots that grow above ground be considered to be?
Interesting question. So on an onion, what about the above ground portion of that plant? If we have roots, stems, and leaves all under the ground, what are those things that are coming out of the top? And the things coming out of the top are actually additional leaves that are connected to the leaves that are formed around the outside of the bulb. Good question. And I'll be the first to say I'm not an onion expert, but I have worked with them enough to know that you lose solids out of them really fast in storage. So I don't know what solids are, but fortunately Taylor has the microphone. And since I asked Spencer an unfair question, I'm joking. Solids are just the carbon or the carbohydrates that are stored in the plant. And when you store, whether it be potatoes or onions or anything else that you're storing that has water in it, it tends to respire a lot and it tends to burn through that carbon pretty quickly. So I'll let you off the hook. That would probably be an unfair question. So moving on to leaves, which should be the last part of what we're doing here. We may actually make this in time today, unless I start getting off on tangents here. Taylor, what's the main purpose of leaves in most plants? All right, the main purpose of leaves in most plants, as Taylor said, is to photosynthesize. So generally speaking, and again, plants specialize themselves to specific environments within which they grow. But by and large, roots are primarily usually going to be responsible for water and nutrient uptake. Stems are primarily going to be important for structure and height and movement of water and nutrients and carbohydrates. And leaves are going to be primarily responsible for the production of carbohydrates through photosynthesis. And leaves, well, go ahead and hand it off. Otherwise, we're not going to give Jeremy another chance to answer a question today. Avery, it is Avery, right? Avery, what do most plant leaves look like? If you had to describe plant leaves, would you say that they tend to look more like a sheet of paper or more like a cylinder?
Probably more
Okay, more flat like a piece of paper. Why is that? Is that just a random chance type of thing?
So they have surface area.
All right, so they have more surface area because surface area is very valuable to a plant for a few different reasons. First of all, it maximizes the ability of the plant to take up CO2 because you've got a lot more pores that can be taking up CO2 without spending a lot of energy in making plant material. It also is useful for protection from excess water loss and also to keep the plant cool. But generally speaking, we'll be spending a lot of time this semester talking about leaves and their function in terms of photosynthesis. Good job, you can hand it over to Caitlin Megan, as opposed to Morgan Megan or Seth Megan. So a typical plant leaf is going to look like this. It's going to have a layer on the top with wax on it. Why do you think it has wax on it? Oh, Alexa and then Caitlin.
I was just wondering, when like a plant is dying or it's sick and it's got no leaves, is it unable to like do photosynthesis?
All right, question was, if a plant is sick and it loses its leaves, is it unable to do photosynthesis? Yes. The stem can still do a little bit of photosynthesis. Any green part of the plant is still technically photosynthetic, but if a plant's losing its leaves, it is signaling to you that it's got bigger problems than the ability to photosynthesize a lot. Generally speaking, you know, shedding of leaves is a last-ditch effort of the plant to try to either keep from losing excessive water or losing excessive other things at the expense of being able to photosynthesize very much. Great question. Okay, so Caitlin, back to my question. The outside of leaves have a waxy layer on them. Why do you think they have that waxy layer, or is that just an accident?
It's to reduce the transpiration.
All right, good. It's to reduce the transpiration. Essentially, the only place that plant wants to lose water is the same place that it's capturing CO2, because it's going to have to lose water just because it's opening itself up to take in CO2. Just like, well, how do we lose most of the water out of our bodies on a daily basis, Caitlin? Okay, yeah, generally speaking, and it depends on the weather, but one of the major pathways for water loss is simply by breathing, because every time you breathe out, you're putting water out of your mouth at about 100% relative humidity. So I guess I should have said major pathways, because there are other major pathways that we use, but why don't we just, to keep from losing as much water, why don't we just hold our breath all day? I realize that's kind of a facetious question, but it's the same issue that plants run into as well. They're going to lose water just because they've got to open their stomata in order to take in CO2, and they're going to lose water as a result. So good job. Go ahead and hand it back to Morgan now. So they protect themselves by, as Caitlin mentioned, having a waxy layer to minimize the water loss from places that they're not bringing in CO2 within the plant. And then immediately underneath, they have these cells. What are all these little green dots inside of these cells, Morgan? And I guess the question could also be, why are plants green? What is it in plants that makes them look green? Okay, chlorophyll. So these little dots in here are chloroplasts, which are the chlorophyll containing molecules in a plant cell. And you see that they're immediately located directly under the surface of the leaf, so that as soon as that CO2 comes in, they've got the mechanism by which they can be doing photosynthesis. Good job. All right. Seth, this is going to be a very difficult question. Do all of these leaves look different? Okay. Are there millions of different leaf shapes in the world? Okay, so everyone remember that, as Seth said, there are millions of different leaf shapes in the world, and the variation allows plants to be identifiable. It also allows plants to have certain adaptations to the environment around them. Good job. Okay, you can hand it off to Grayson now. Okay, Grace, another difficult question. There are a lot of different types of leaves. You can have simple leaves, you can have various types of compound leaves. So this is actually a philosophical question. Grayson, do you think it's important for the class to memorize all of the different types of compound leaves for a crop physiology class? Okay, Grayson has just told you that you don't need to memorize the different types of leaves on this page. So everyone thank Grayson. Otherwise, I was totally going to ask you like 3 questions on the exam about the different types of compound leaves. So don't worry about memorizing the details on this. Just be aware that leaves come in a lot of different shapes and sizes, and you know there's an entire branch of plant anatomy that focuses on the difference between, for instance, compound leaves and compound leaflets, which are parts of a leaf. So you can have different leaves and you can also have different parts of a leaf, and you can't tell the difference except as you get in with a microscope and see how they're connected with petioles or not. So just be aware that there are different types. Good. Okay. Structure. I may ask you this simply because I've asked this in the past, but what is that little stem type protrusion that connects a plant leaf to the plant stem? You know, Carson? Looking at this leaf right here, we've got what's definitely the leaf, and then we've got the stalk that connects the leaf to the rest of the plant here. What is the stalk called right here? Okay, good. This is the petiole. So interestingly enough, the petiole is defined as a modified stem as opposed to being defined as a leaf. I don't know why they do that, but when we're taking samples, we actually separate the leaf tissue from the petioles, and we test them for nutrients separately because the petiole's connective tissue and the leaf is considered the photosynthetic portion. So just be aware of that. If you don't have a petiole, these leaves are actually called sessile leaves. So if you see the term sessile, just be aware that that's a leaf that doesn't have a petiole. I probably won't ask you that, but I have asked that in the past, and I'm not above going through and rating old questions. So, you know, just make a note of that, you know, a sessile leaf is a leaf without a petiole, and if it pops up on the exam, you'll be prepared. We also have something really cool. Some types of plants that form rosette, they will actually do something unusual, which is, let's see if I can jump to it right quick here. A rosette type plant is actually going to design itself to maximize light absorption by changing the petiole length. So a rosette type plant is going to have a long petiole at the bottom, long petioles at the bottom, and then as it gets up higher, it's going to have shorter and shorter petioles. And so each of these leaves is essentially capturing sunlight in its own zone here. And so if you see a rosette type plant, yes.
Would a Chinese money plant be that kind of plant?
Question, would a Chinese money plant be that type of plant? Well, let's see what a Chinese money plant is. I'll admit, I'm not very up on my.
The circular, yeah. Circular.
My answer would be probably, except it's kind of difficult to tell on this one. These, I guess, are they all connected at one base? Then probably so. Have a good one, Rogelio. Sorry, I didn't get to you for your last question. Generally speaking, if you see a plant that spreads out into a nice rosette, such as potentially a Chinese money plant, assuming I'm not wrong, it's going to be doing that by changing the length of the petiole and to some extent changing the length of the individual leaves as well. Some of the other adaptations, if you've got plants that have an advantage of, sometimes plants adapt themselves to lower light environments. And they may do this for a number of reasons. Generally, it's to be more competitive when there's not as much sunlight. They may actually reflect light. They may use leaf hairs to repel insects, to decrease air movement and transpiration. A lot of your, let's see, who has the microphone? All right, George. So, decreased surface to volume ratio, such as succulents and cacti. You tend to see aloe vera plants and cacti in a very wet environment or more in a very dry environment. Okay, you see them in a very dry environment. And when you look at them, they have huge layers of wax on them, first of all. And second, they tend to be a lot heavier leaves than you get in a typical plant because they've adapted themselves to photosynthesize by capturing CO2 at night, so they don't have to have their stomata open. And then during the day, their main goal is to just keep from getting any hotter than they absolutely have to. So great answer on that. All right, you're gonna have to walk it all the way over to Calvin here. On the bright side, we're down to like the last 3 slides here. What do you need to know about this slide? Not a whole lot. I'm sure you've seen this in your introductory biology classes, and we'll talk about some of these things in more detail later, but just be aware that if you were to slice a leaf open, you're going to see vascular tissue, which is moving carbohydrates out and water and nutrients in. You're going to see mesophyll cells and bundle sheath cells that are going to be responsible for photosynthesis. You're going to see a cuticle that's waxy. You're going to see epidermal cells. You're going to see guard cells, etc. So Calvin, I feel like this is too easy of a question as well, so I'm sorry. Way back to the beginning of the lecture today, what type of cells would you call these cells? Do you think these would be undifferentiated cells or differentiated cells? Differentiated, yeah. I'm sorry. I'll ask you a more difficult question next time, but it's a great way to wrap it up. So, generally speaking, your leaves are going to be initiated at the apical meristems of stems or branches as leaf primordia, which are going to be differentiated or undifferentiated. Yeah. Undifferentiated. So they start out as undifferentiated. Then over time, they differentiate out into cool things like vascular tissue and epidermal tissue and etc. In monocots, the meristems are actually going to be located at the base or the crown of the plant. Some dicots are going to do that to some extent, but generally you see that with monocots, and usually the dicots, the meristems are going to be up around the margins of the leaves and the top of the plant. So last slide. Why do you think I'm even showing this slide, Enika? All right, to remind you of what we just spent the last hour and a half talking about. And also to remind myself when I'm writing questions for the exam, what I talked about today. So make sure that you're comfortable with these different processes. Make sure you're familiar with different types of meristems. Make sure that you're familiar with the basic functions of roots, stems, and leaves in a plant. Make sure that you know a few types of specialized stems in particular, and that should do it. Everyone, there's a winter storm advisory for this evening, so if there's ice on the road, drive really slow in the morning, because Amarillo and Lubbock are the two places that I have run into the most slippery roads that I've ever seen. And I've driven on a lot of ice in my life. Other than that, have a great afternoon, and I'll look forward to seeing you on Thursday. Yes.
This is my first ever, and so I'm figuring it out because I want to be a soil sciences minor, and
Okay.
so I just want to let you know
No,
that
that's great.
if you
And-
get emails from me
Sure.
or whatever,
No,
I
and always feel free to reach out. I've had quite a few students over the years take this as freshmen or, early in their career before they've had a lot of other science classes. They've done fine. And this goes for everyone. If you ever have any questions, or if you feel like you're getting behind on something, don't ever hesitate to reach out, because this is a class that if you stick with it, you'll be able to stay up with. You don't strictly need any prerequisite in order to be successful. Helps if you're able to do a little bit of math, but again.
yeah. Okay,
All right.
and then